In this lesson, we're going to learn how the genetic code gets translated into proteins and other molecules. Now, the first thing we have to understand is what a codon is. We're going to see this word a lot. A codon consists of 3 consecutive bases, and they specify a single amino acid that is to be added to the polypeptide chain during this process we call translation. Now, all living organisms read the genetic code the same way, using these 3 bases at a time. So, what are the 3 letter groups of codons shown here? Well, you can see we have the AUG codon, we have an AAC codon, and finally a UCU codon. Now, a typical mRNA strand that codes for a specific protein is going to be much longer than this, but we're just using these 3 as an example, these 3 codons. So only 9 bases. Now, we have the advantage of having what's called a genetic code table. And this actual table helps us to identify which amino acids are coded for each specific codon. So since there are 4 different bases in RNA, that means there are a possible 64 combinations of these 3 base codons. Okay, you figure that out because there's 4 letters: A, U, C, and G, and we use 3 in a codon, a 3-base codon, so that would be 4 times 4 times 4, and that equals 64. That's how we get that. Okay. So reading those codons and reading the amino acids that are translated by those codons is simplified by using one of these tables that you see here. And you'll have access to this for any type of assessment that you might have to answer questions about codons specifying particular amino acids. Now, most amino acids can be specified by more than one codon. For example, there are 6 codons, which are UUA, UUG, CUU, CUC, CUA, and CUG. They all specify for leucine. In fact, here are a couple of those. So you see UUA and UUG right here. So you read from the inside out, by the way, if I didn't mention that. You go from U to A, and we get leucine. Or you go U to G, you get leucine as well. Okay, so UUA and UUG. Okay, we also can get leucine over here. You see leucine. It's coded with CUU or CUC or CUA, and finally CUG also codes for leucine. And you'll find that there are a number of amino acids that actually have multiple codons that code for them. In fact, almost every single one of them have at least 2. There are a few that only have 1. For instance, methionine, which is AUG, that only has 1 codon, but that's a very specific codon we're going to learn about here in a few minutes. But we also have, for instance, only 1 codon for tryptophan. UGG is the only codon that codes for tryptophan, the amino acid tryptophan. Now, although the code table that we have here applies to most multicellular organisms, including animals and plants, there are some slight differences in the code as used by some of the microorganisms. Also, some of the mitochondria and chloroplasts also use a little bit of different code. All right, so we're going to read and decode a couple codons. So we're going to use a codon of C, A, C. That's going to be our codon that we're going to use. Okay. Now, how do we find the amino acid that is translated by CAC? Well, we start in the center with C. And then we work our way outwards. So the next letter would be A. So anywhere inside of the area that C covers, the inside C, see how there's a line here that separates the 4 letters on the inside? Okay. There will be 4 letters for each of the first letters. In the second level, there'll be 4 letters, and in the third level, there could be a variety of different numbers of letters. But what we have is C, U, and then all 4 of the letters can be coded for as the third letter in the codon. But we're looking specifically for CAC and the amino acid that it codes for. So we start with C, we work our way out to the next one, which is one of these has to be A, and of course, there it is right in front of you, right below the C there, off to the right a little bit. And then our next letter is C, so within the A region, which can be any 4 of these letters, we find C, and we see that there. And that means that we are now in the section for histidine. Okay, so histidine is what CAC codes for. Okay, now CAU would also code for histidine, but in this case we were translating the codon CAC. That translates into histidine. Okay. Boom. CAC equals histidine. Now, I told you a moment ago about a codon called methionine, and it's right down here. And it is a special codon. It is AUG. Notice that is the only codon that will code for methionine. It's a special codon because it tells the ribosomes to start coding for the protein. And then there's also some other codons that are the stop codons. So methionine is going to be AUG, and that is the start codon. So if I say, what's the start codon? You should be able to tell me it's methionine. If I say, what is the codon for, well, actually, I guess if I say, what's the start codon, you would say AUG. If I say, what's the start amino acid, you would say methionine. Okay. Now, following the start codon, the mRNA is going to be read 3 bases at a time until it reaches one of 3 different stop codons. So instead of a start codon like methionine or AUG, we are now talking about stop codons that actually end translation. So let's see if we can find some of these stop codons. Let's find all 3 of them. All right, the first one is UAA. I was looking at the A. UAA is a stop codon. Okay. Now we can simply change the A, the third A, to a G, And again, it's still the stop codon. Okay. And there is one other stop codon you might notice right here, and that is AGA. So UAG, UAA, and UGA all are stop codons. UAA, UAG, and UGA. All right, so translation actually occurs after transcription. So transcription is the process of DNA being copied to make mRNA strands. Or RNA, and then when it leaves the nucleus, it is then considered mRNA. And it's mRNA that directs the translation process. So here in this illustration, we have our strip of mRNA. It's coming out of the nucleus, as you can see. Of course, this has got to be a eukaryotic cell in that case. The fact that there's a nucleus there indicates that this is a eukaryotic cell. And we have to keep in mind that transcription is what takes place inside of the nucleus. Translation is the process that ribosomes use to transcribe the codons of the mRNA strand into proteins or other molecules. And this occurs in the cell's cytoplasm. It doesn't occur inside the nucleus. So translation starts when the ribosome actually attaches to one of those mRNA molecules in the cytoplasm. And again, just like we talked about, it initiates at the AUG codon, the start codon. And in this illustration, you see it right there, AUG, a little off center, but you get the idea. Now, tRNA molecules come into play here as well. And these are the molecules that carry those amino acids that are coded for by the codon. And they are complementary to the mRNA strand as well. Now, what do I mean by they are complementary? Well, you can see right here that there are 3 U's in a row. And here is 3 A's in a row. So the codon AAA, if we go back to our wheel, we see that AAA is lysine. So AAA is going to code for lysine. Now notice that the codons that are attached to the tRNA are the complement to the codons on the mRNA strand. So the tRNA molecule that carries lysine has the complementary bases of UUU. Now down here we have UUC. So if we jump to our wheel and we go UUC, UUC is phenylalanine. UUC gives us phenylalanine. Now, again, the tRNA molecules are going to carry those amino acids that are coded for by the codon on the mRNA. And since AUG always codes for methionine, the first amino acid brought in for every round of translation is always methionine. And we see that here. Methionine is brought into the AUG. Now, as each codon passes through the ribosome, more tRNAs are going to bring in the proper amino acids into the ribosome. And here we have a lysine being brought in, which we kind of already talked about. Now, each transfer RNA, each tRNA, has what we call an anticodon. So it's the complementary codon to the codon that's drawing it in. But we don't call that a complementary codon. We call it an anticodon. So we have codons on the mRNA and we have anticodons on the tRNA. And those anticodons are going to match up with their complementary codons on the mRNA. Okay, once that attaches, then the ribosome then creates a bond between the amino acids. So it keeps adding more and more amino acids until it gets to the end to a stop codon, and then it stops adding protein or amino acids at that point, and it is now considered a particular protein, depending on the string of amino acids that have been added. So there's the anticodon right there. That's going to be a part of every tRNA molecule. All right, so the ribosome helps form this peptide bond between the first and second amino acids as it brings it in. So this would be between methionine, and in this case, the second one is phenylalanine, if you remember from the previous slide. And we see that right there. So you see a small mark, a little line between the two amino acids that indicates that a bond has been formed. So the ribosome's job is to not only bring in the correct tRNA, the correct anticodon, but is also going to create a chemical bond between those amino acids. Now, at the same time, the bond that's holding the first tRNA molecule to its amino acid, that bond actually gets broken. And the tRNA then moves into a third binding site from which it exits the ribosome. Okay, so that very first tRNA enters a third area, if you will, where the bonds are broken and it exits the ribosome. So as the ribosome keeps moving to the next codon, the next codon, the next codon, 3 codons in front of that, those codons, those tRNA molecules are being released. Now, they're being released without their amino acid, so they're going to have to go pick up an amino acid, and only certain amino acids will attach to the tRNAs based on the anticodon that tRNA is carrying. Okay, so the ribosome just continues moving along the mRNA molecule from right to left, binding new tRNA molecules and amino acids as it goes. Now, keep in mind that the overall picture and relationship between transcription and translation is transcription is an exact copy of a section of the DNA, right? So we're going from nitrogenous base to nitrogenous base. Translation is kind of like language, where you translate, say, from English into Spanish or something like that, right? So we're not going from nitrogenous base to nitrogenous base. Now we're going from nitrogenous bases to amino acids. So we call this translation. So what is the product of transcription that happens inside the nucleus? The product of transcription is mRNA, but the product of translation is a protein. So very similar in some ways, but also very different. The end result is extremely different. So where do the amino acids come from that make up the protein? Well, they're available, they're floating around in the cell, and they're picked up by those tRNA molecules based on the anticodon that tRNA molecule is carrying. And the tRNA molecules then bring them into the ribosome to be used to build these proteins. So the translation process continues until the ribosome reaches one of those 3 stop codons that we looked at earlier. So we see that there is a stop codon at the end of this mRNA strand in this illustration, a short strand for sure, but you see it right here. UGA is one of those stop codons. So when the ribosome reaches the stop codon, it basically releases both the newly synthesized polypeptide, and it also releases the mRNA molecule, and at that point, the process of translation is complete. Now, here we see the polypeptide in the top, a long string of amino acids that are all chemically bonded to each other to form a protein. And here, of course, the bottom is the mRNA strand that was used to make that polypeptide. So what would happen if the stop codon mistakenly had been made into a regular codon? Well, if you're thinking it wouldn't stop the translation process, you are correct. The translation process would continue. Another amino acid would be attached to the protein product, and we would end up with a protein that doesn't function properly. There'd be an error in its structure. Or we might end up with a different protein altogether. So all three major forms of RNA, which are mRNA, tRNA, and rRNA, are involved in the process of translation. So we'll start with mRNA, messenger RNA. This is the molecule that carries the coded message that directs that process of translation. The tRNA, its role is to deliver the amino acids, which enables the ribosome to translate the mRNA's message into protein form. And then finally, we have ribosomal RNA, which we haven't really talked about a lot, but essentially the ribosomal RNA molecules hold ribosomal proteins in place and might even carry out the chemical reaction that joins those amino acids together. We're still learning about how all this works. So all of these components, they translate the genetic code for the purpose of synthesizing new protein molecules, and this is common to all organisms. We have DNA to RNA to protein. That is essentially the process, or what we would call the central dogma of molecular genetics. We go from DNA to RNA, that is transcription. Hard to read when it goes crazy like that. Transcription. It's a long word. Transcription, and this is translation. Okay? Hard to read, but I think you get the idea. Transcription is DNA to RNA. Translation is RNA to protein. Okay? This is what represents the central dogma of molecular biology. So what does the central dogma imply about the role of RNA? Well, it essentially is the step in between DNA and proteins. So the central dogma does have some limitations. For example, it doesn't it doesn't represent the other roles of RNA. There are also some exceptions to this dogma, including viruses that transfer information in the opposite direction from RNA to DNA. Okay, that's what happens when there's a viral infection in a cell. Now, let's take a look at this illustration. There's a gene expression involved in this particular illustration. Consider the way in which the DNA, RNA, and proteins are involved in putting genetic information into action in a living cell. And also keep in mind, though, that this illustration really is a simplification of the process. So the protein that gets made at the end of transcription and translation is the ultimate use or expression of the code in the DNA segment that was used in transcription. Scientists would say that that region of DNA has been expressed. This means basically that its code was used to build a protein. So DNA carries information for specifying the traits of an organism. The cell uses the sequence of bases in DNA as a template for making mRNA, and then the codons of the mRNA specify the sequence of amino acids in a protein. Proteins, in turn, play a very key role in producing an organism's traits. So a lot of RNA molecules are not even translated into proteins, but they still play important roles in gene expression. Now, this diagram is, you can see the relationship of it to the central dogma of molecular biology. So we start with the transcription of DNA into mRNA. And then we see the mRNA being translated into a polypeptide chain. Now, one of the most interesting discoveries of molecular biology is the near universal nature of the genetic code. Now, some organisms do show some slight variations in the amino acids assigned to particular codons, but the code is always read 3 bases at a time and in the same direction. Despite their enormous diversity in form and function, living organisms display a very remarkable unity at life's most basic level. And this is the molecular biology of the gene.